For decades, scientists have searched for the earliest traces of human life, digging through caves, ice, and soil, piecing together fragments of bones and tools. Until recently, the oldest human DNA remained elusive, breaking or degrading over thousands of years. Now, in a groundbreaking discovery, researchers have successfully sequenced DNA from human remains that date back over 400,000 years. This is not just a technical achievement, it is a window into our deepest past, revealing the origins, migrations, and genetic diversity of early humans. The findings challenge long-held assumptions about human evolution, revealing unexpected connections between ancient populations in Europe, Africa, and Asia. Today, we calmly explore these discoveries, uncovering what the oldest human DNA tells us about who we are, where we come from, and how the story of humanity has been far more complex than anyone imagined. The origins of the search for ancient DNA. The search for ancient human DNA began in the late 20th century when scientists first attempted to extract genetic material from old bones and teeth. Early attempts were often unsuccessful because DNA degrades rapidly after death. Heat, moisture, and microbial activity break down genetic material, leaving only fragments that are difficult to analyze. Despite these challenges, researchers remained determined, developing techniques to isolate and amplify the tiny pieces of DNA that remained. Advances in polymerase chain reaction technology allowed scientists to recreate sequences from minimal samples, revolutionizing the study of ancient genetics. Each discovery opened new doors into understanding human evolution, migration, and adaptation. The sequencing of Neanderthal DNA in the 1990s was a major breakthrough, showing that humans had interbred with other species tens of thousands of years ago. These successes inspired a new generation of paleogeneticists to look even further back in time, targeting specimens hundreds of thousands of years old. By carefully selecting bones from cold or dry environments, scientists maximized the chances of preserving DNA. This long-term effort laid the foundation for the sequencing of the oldest human DNA ever recovered, a project that would take decades of research, patience, and innovation. The discovery of the 400,000-year-old human remains. The breakthrough came from a cave system in Spain, where fossilized human remains had been preserved in unusually stable conditions. The bones belonged to an early human species known as Homo heidelbergensis, considered a direct ancestor of both Neanderthals and modern humans. Researchers were initially skeptical that DNA could survive for 400,000 years, but the cold, dry cave environment slowed decomposition, protecting microscopic traces of genetic material. After careful extraction, sequencing, and analysis, the DNA revealed a remarkable picture of ancient human diversity. What was most surprising was that the genetic material had enough integrity to allow comparisons with Neanderthal, Denisovan, and modern human genomes. This meant that scientists could trace evolutionary relationships across hundreds of thousands of years, reconstructing the movements, interbreeding events, and adaptations of early humans. The find also challenged assumptions about the survival of DNA, showing that under the right conditions, genetic material can persist far longer than previously believed. This discovery not only pushes back the timeline of genetic analysis, but also opens the possibility of exploring even older specimens in the future. Homo heidelbergensis and its place in human evolution. Homo heidelbergensis is a critical species in human evolution, serving as a bridge between Homo erectus and both Neanderthals and modern humans. Fossil evidence suggests that they were large-bodied, highly adaptable, and capable of using sophisticated tools. They lived across Europe and Africa between 600,000 and 200,000 years ago, occupying diverse environments from forests to plains. The new DNA sequences confirm that these populations were genetically diverse, more than previously thought. This species likely contributed genetic material to later populations, influencing traits seen in Neanderthals and modern humans. The findings also show that the evolutionary tree is not a straight line, but a complex network of branching and interbreeding populations. Homo heidelbergensis appears to have experienced periods of isolation followed by contact with neighboring groups, leading to genetic exchange. By analyzing ancient DNA, scientists can identify genes associated with physical traits, disease resistance, and adaptation to climate, revealing the challenges early humans faced and how they overcame them. This research deepens our understanding of human ancestry, showing that the story of evolution is more intricate than textbooks often present. Techniques that made this discovery possible. Sequencing ancient DNA is one of the most difficult tasks in modern science. The genetic material is often fragmented into tiny pieces, contaminated with bacteria, fungi, and modern human DNA. To overcome this, researchers developed specialized techniques, including next-generation sequencing, which can read millions of DNA fragments simultaneously. Careful laboratory protocols prevent contamination, while bioinformatics tools reconstruct the fragmented sequences into complete genomes. In the case of the 400,000-year-old DNA, scientists used bone cement extraction, a method that targets the densest part of bones where DNA is most likely preserved. Chemical treatments were applied to remove contaminating DNA, and machine learning algorithms helped identify authentic ancient sequences. The process is painstaking, often taking years of trial and error. Each successful read of DNA adds a piece to the puzzle, revealing mutations, lineage markers, and genetic variations. Without these innovations, the oldest human DNA would have remained inaccessible, leaving questions about ancient evolution unanswered. This breakthrough is a testament to human ingenuity, showing that we can overcome the limits imposed by time, decay, and environmental conditions to peer into our distant past. What the DNA reveals about interbreeding. One of the most shocking revelations from the oldest human DNA is evidence of interbreeding between ancient populations. Homo heidelbergensis, Neanderthals, Denisovans, and early Homo sapiens were not completely separate species. Instead, they frequently interacted, shared genes, and influenced each other's evolution. The new DNA sequences show that early European populations had genetic connections to Neanderthals long before previously thought. Certain genes responsible for immunity, skin color, and brain development may have been passed between these groups, shaping traits in both modern humans and their ancient relatives. This challenges the old idea that species evolved independently without interaction. Instead, human evolution is more like a tangled web of relationships, with migration, isolation, and interbreeding all playing critical roles. These findings also help explain why modern humans carry small percentages of Neanderthal and Denisovan DNA in their genomes. The oldest DNA provides the earliest evidence of these exchanges, pushing back the timeline and showing that interbreeding has been a fundamental part of human survival for hundreds of thousands of years. Insights into ancient human migration. The sequencing of the oldest human DNA provides unprecedented insights into how early humans migrated across continents. Before this discovery, scientists relied primarily on fossil evidence and stone tools to map migration patterns, but these sources offered limited detail. DNA reveals not just where humans traveled, but also when and how populations interacted along the way. Analysis of the 400,000-year-old genome shows that Homo heidelbergensis populations in Europe were genetically distinct from African counterparts, yet there were periods of contact and gene flow. This suggests that early humans did not migrate in straight lines or remain completely isolated. Instead, they moved intermittently, influenced by climate, availability of resources, and competition with other species. Ice ages, sea level changes, and shifting landscapes likely shaped these movements, creating corridors and barriers that determined where humans could live. These migrations were not random. They reflect adaptability, strategy, and resilience in the face of environmental challenges. By mapping genetic markers, scientists can reconstruct these ancient journeys with surprising accuracy. For example, certain genetic traits found in the European Homo heidelbergensis population suggest that they carried adaptations for colder climates, indicating seasonal migration or long-term habitation in northern regions. This DNA-based approach revolutionizes our understanding of human dispersal. It shows that migration has always been central to survival and evolution far earlier than previously documented. Humans were explorers from the very beginning, moving across the continents long before modern Homo sapiens emerged. Clues about early human health and immunity. Ancient DNA also reveals information about the health, disease resistance, and immune systems of early humans. Certain genes preserved in the 400,000-year-old genome indicate that Homo heidelbergensis had adaptations to fight pathogens common in prehistoric environments. For instance, genetic markers associated with resistance to bacterial infections and parasites were present, showing that even ancient humans faced pressures from disease. Studying these sequences helps scientists understand how immunity evolved over hundreds of thousands of years and why some genetic traits persist in modern populations. Remarkably, some alleles found in the ancient DNA are still present today, inherited through interbreeding with later human species. This continuity demonstrates that natural selection favored genes providing survival advantages. Additionally, other DNA segments provide insight into diet and metabolism. Genes related to digestion suggest that early humans consumed diverse diets, including plant materials, meat, and possibly underground tubers. These findings align with archaeological evidence of stone tools used for hunting and processing food. The DNA also reveals information about stress responses, growth, and longevity, painting a picture of how humans adapted physiologically to environmental pressures. Overall, the oldest human DNA offers a window into the daily survival challenges faced by our ancestors, showing that evolution shaped not only physical features, but also complex internal mechanisms like immunity. Surprising links to Neanderthals and Denisovans. The sequencing of this ancient DNA uncovered unexpected connections to Neanderthals and Denisovans. Prior to this discovery, scientists assumed that Homo heidelbergensis was an isolated branch, but the genetic data tells a more intricate story. Certain gene sequences from the 400,000-year-old human show clear similarities with Neanderthals, suggesting that the European populations of Homo heidelbergensis were likely ancestral to later Neanderthal populations. Surprisingly, there are also traces of genetic features linked to Denisovans, hinting at ancient interactions that were previously unknown. This demonstrates that early human species were not completely separate. They shared DNA, exchanged genes, and contributed to each other's evolutionary paths. These findings challenge the traditional tree-like model of human evolution, which depicted species as distinct branches. Instead, evolution appears more like a network, with multiple species interacting over long periods. These ancient connections also explain why modern humans carry small amounts of Neanderthal and Denisovan DNA. Certain traits, such as adaptation to cold climates, disease resistance, and even aspects of brain development, were likely inherited from these ancient encounters. The sequencing of this oldest DNA effectively rewrites our understanding of human ancestry, showing that our evolutionary history is far more interconnected than previously thought. Insights into human cognitive evolution. Ancient DNA also provides clues about the evolution of the human brain and cognition. While fossils can reveal skull size and shape, genetic analysis allows scientists to examine genes associated with neural development, learning, and sensory perception. The 400,000-year-old DNA contains sequences linked to brain growth, suggesting that Homo heidelbergensis already possessed cognitive abilities more advanced than earlier species. These genes hint at complex behaviors, including tool use, social structures, and possibly symbolic thought. Archaeological evidence of hunting strategies, organized camps, and coordinated efforts aligns with these genetic findings, indicating that these early humans had sophisticated planning and problem-solving skills. Furthermore, some genetic markers related to speech and communication appear to have been emerging during this period. While it is impossible to know exactly how language sounded, the DNA suggests that neural pathways supporting complex vocalization may have been developing. This challenges the notion that cognitive complexity is unique to modern humans. Instead, it shows that advanced thinking and social behaviors were evolving gradually, influenced by both genetic adaptation and environmental pressures. The oldest DNA provides a remarkable opportunity to trace the roots of what makes humans uniquely intelligent, bridging the gap between primitive behavior and modern cognitive abilities. What the DNA tells us about physical traits beyond behavior and cognition. The 400,000-year-old DNA reveals physical traits of ancient humans. Genetic analysis indicates that Homo heidelbergensis had dark skin, likely an adaptation to lower ultraviolet radiation levels in Europe at the time. Hair texture, eye color, and facial structure can also be inferred from specific gene sequences. For instance, some sequences suggest strong jawbones, large teeth, and a robust skeletal structure adapted for strenuous activity and cold climates. These traits align with fossil evidence showing heavily muscled bodies capable of long-distance travel and hunting. Interestingly, certain genes associated with height, bone density, and immune system strength are remarkably similar to those found in modern humans. This demonstrates continuity and evolutionary pressures that shaped humans over hundreds of thousands of years. These physical insights help scientists reconstruct a realistic image of our ancestors, moving beyond skeletons to living, breathing humans with adaptations shaped by climate, diet, and environment. It is a vivid reminder that the oldest DNA is not just a scientific curiosity, it is a direct link to the people who walked the earth long before modern civilization existed. Clues about diet and environment. The oldest human DNA also provides remarkable insights into the diet and environment of early humans. Genetic sequences reveal adaptations for processing a diverse range of foods, from protein-rich meat to fibrous plant matter. Enzymes associated with starch digestion suggest that early humans consumed tubers, seeds, and grains, even hundreds of thousands of years ago. Other genetic markers indicate a capacity to metabolize fats from large game, which aligns with archaeological evidence of hunting tools and butchered animal bones. Environmental adaptations are also visible in the DNA. Genes linked to cold tolerance suggest that European populations of Homo heidelbergensis were evolving traits necessary to survive Ice Age conditions. Similarly, variants related to vitamin D synthesis indicate responses to lower sunlight levels in northern latitudes. By studying these genes, scientists can reconstruct the challenges faced by early humans, including climate, seasonal food scarcity, and exposure to pathogens. This level of detail allows a comprehensive view of daily life for ancient humans, far beyond what fossils alone could provide. The DNA tells us that survival required flexibility, adaptability, and innovation, traits that continue to define humanity today. What it reveals about longevity and aging. Ancient DNA also sheds light on the lifespan and aging process of early humans. While direct fossil evidence can suggest average ages, genetic analysis provides clues about cellular repair mechanisms, susceptibility to disease, and longevity-related traits. Some sequences indicate genes for efficient DNA repair and resistance to oxidative stress, which may have helped Homo heidelbergensis survive in challenging environments. Other genes show vulnerabilities to infection and injury, highlighting the harsh conditions faced daily. Studying these traits in combination with fossil records allows scientists to estimate life expectancy and physical resilience. It appears that early humans faced high mortality rates, particularly during childhood, but adults could live into their 40s or 50s under favorable conditions. These genetic insights help contextualize archaeological findings, showing how environmental pressures and evolutionary adaptations shaped not only survival but the human life cycle. The oldest DNA emphasizes that aging and longevity have long been influenced by a combination of genetics and environment, a relationship that continues to play a crucial role in modern human health. The implications for human evolutionary models. The sequencing of 400,000-year-old DNA challenges traditional models of human evolution. Previously, scientists imagined a linear progression from Homo erectus to Homo sapiens, with Neanderthals and Denisovans as separate branches. The new genetic evidence reveals a much more complex picture, with frequent interbreeding, migrations, and gene flow between populations. Homo heidelbergensis appears as a critical connector, contributing to multiple lineages simultaneously. This interconnectedness implies that traits such as cognition, immunity, and physical adaptations were shared across populations, rather than evolving in isolation. Evolution is therefore not a straight line but a tangled network of interactions influenced by climate, geography, and social behavior. The oldest DNA allows scientists to rewrite textbooks, providing a more accurate depiction of human history. It shows that the origins of modern humans involve contributions from multiple ancient species, and that diversity has always been a driving force in our evolution. Insights into disease and immunity. The oldest DNA also offers a rare window into how early humans coped with disease. Genes linked to immune response indicate that Homo heidelbergensis was already adapting to pathogens present in their environments. Some alleles show resistance to bacterial infections, while others suggest exposure to parasites common in prehistoric times. By studying these sequences, researchers can trace the origins of certain diseases and immune system adaptations that persist in modern humans. These findings also show how natural selection shaped the human genome over hundreds of thousands of years. Early humans faced constant challenges from their environment, and immunity was critical to survival. Understanding how ancient DNA reflects these pressures helps scientists learn not only about past populations, but also about the genetic foundations of disease resistance today. The role of climate and geography. Climate and geography shape the evolution and survival of early humans in profound ways. The sequencing of 400,000-year-old DNA shows that Homo heidelbergensis populations had genetic adaptations for cold European winters, including efficient thermoregulation and metabolic flexibility. Fluctuating temperatures, ice ages, and seasonal variations in food availability exerted constant pressure on these populations, influencing migration patterns and survival strategies. Rivers, mountains, and natural barriers dictated movement, creating isolated populations that developed unique genetic traits over time. At the same time, changing climates created opportunities for contact between previously separated groups, resulting in interbreeding and the exchange of genetic material. These environmental pressures not only shaped physical traits, but also influenced behaviors, tool use, and social organization. The DNA reveals evidence of genetic markers linked to sunlight exposure and vitamin D synthesis, reflecting adaptation to limited sunlight in northern latitudes. By understanding these connections between genes, geography, and climate, scientists can reconstruct the challenges faced by ancient humans and the ways they overcame them. This research shows that adaptation was not just about survival, it was a dynamic process influenced by constantly shifting landscapes and climates. The story of human evolution is inseparable from the environment, and ancient DNA provides a rare window into how early humans responded to the world around them. Lessons for modern humans. The discovery of the oldest human DNA carries important lessons for modern humanity. First, it underscores resilience. Homo heidelbergensis survived extreme climates, scarcity of food, and exposure to pathogens, adapting both genetically and culturally. Second, it highlights the interconnectedness of all humans. The tangled web of ancient interbreeding revealed by the DNA demonstrates that modern populations share ancestry with multiple extinct species, reminding us that diversity has always been central to human survival. Third, the findings emphasize the power of careful scientific inquiry. By analyzing tiny fragments of DNA preserved for hundreds of thousands of years, scientists have rewritten the story of human evolution, migration, and adaptation. These lessons are not only historical, they inform how humans confront challenges today. Climate change, disease, and resource scarcity are modern parallels to the pressures faced by early humans. Studying ancient DNA encourages us to appreciate the adaptability of our species and to recognize that progress comes from learning, cooperation, and innovation. The oldest DNA also reminds us that humanity is part of a continuous journey, shaped by countless generations, each leaving a legacy encoded in our genes. It inspires reflection on how we can use knowledge from the past to navigate the future. What the oldest DNA ultimately reveals at its core, the oldest human DNA, tells a story of survival, adaptation, and connection. It proves that early humans were intelligent, resilient, and capable of complex behaviors such as tool making, cooperation, and planning. The DNA reveals that human evolution is not linear, but a network of populations exchanging genes, adapting to climates, and learning from one another. It provides evidence of migration patterns, physical adaptations, cognitive development, and immune system evolution. Most importantly, it shows that the roots of modern humans are deeply intertwined with multiple ancient species,